so what we're really interested in right now is the aging was surprising, seeing how aged microglia and astrocytes, what makes them not work well. Why are there dead cells in the brain? I think that is important because aging is one of these things that everyone ages. So so it's a very it's part of life. But so what we're planning on doing now is to understand the molecular mechanisms of how that interaction between astrocytes and microglia are in an aged brain. Like what is, and compare it to the young brain and say, where has this gone wrong? And why do you have debris laying in the brain? Like where can we understand the interactions between astrocytes and microglia, and also their interaction with a dying cell, and independently and together, and see how we can, the molecular mechanisms there, and see how we can intervene during aging. And so that's where we're really kind of working on to understand the molecular mechanisms that are dysfunctional in the aged brain. Yeah. So the beauty of two fatal is that it's one single cell. You can choose the cell to kill the cell. You have no surrounding tissue injury, which I think is a beautiful technique. However, when you think about trauma to the brain, it's thousands of cells that die. And so the beauty of two fatal is that it gives you this very good spatiotemporal resolution of what's happening, but that may, as more and more cells die, that may no longer be applicable. So the response that astrocytes and microglia may have to one dying cell is probably different from what it would have to a population of 100 cells, for example. And so what we're trying to do is try to expand the number of cells actually that you can actually delete or at the same time, and see, okay, during injury or during neurodegeneration, what happens at that point. So we're trying to really expand that method and even look at subcellular, so not just the cell itself, but inside the cell, like what are the different processes that occur inside the cell that lead to cell death. So